on this computer. Perfect. All right, hello. Welcome to our joint Montana Tech public lecture series, as well as Earth Science and Engineering seminar. So we have our second Earth Science and Engineering speaker. We're very pleased to have Karen Dannemiller from the Ohio State University today with us. And we have our flyer up here. So from our flyer, Karen directs the Indoor Environmental Quality Research Group, and she looks at emerging environmental concerns within the built environment. She's an assistant professor at the Ohio State University with a joint appointment in civil, environmental, and geodetic engineering and environmental health sciences. She's a core faculty member of Sustainability Institute, as well as a courtesy appointment in microbiology. So her work looks at improving the understanding of chemical and microbial processes indoors that impact health while fostering student engagement. And she has funding from all sorts of places, including NSF, NIH, NASA, HUD, Alfred P. Sloan, as well as Sochi AFRL. And recently, she has a National NSF Career Award, Buckeye Engineering Women in Executive Leadership, an Innovation Award, and the Lumley Engineering Research Award. So we are very pleased to have Karen speaking with us today via Zoom, and maybe we'll have her in person sometime. About the indoor microbiome, novel applications in mold prevention, healthy spacecraft, and COVID-19. Okay.
Great. Thank you so much for the introduction, and thank you so much for having me today. I'm really excited to be there. I'd love to come see beautiful Montana someday. That sounds great, and I'd be happy to come meet all of you in person. Let me share my screen. Okay. So today I'm going to talk with you all about our work on the indoor microbiome and specifically some applications that we've been working on, particularly mold prevention, design of healthy spacecraft, as well as COVID-19 surveillance. I like to start with this slide because it shows the International Space Station, which is something that we're looking at, the microbiome of the International Space Station. And just to remind everybody that this is something you can actually view on your own. You can just go to spotthestation.nasa.gov and check it out. I did check viewing opportunities in Butte. There's not anything right now, but if you check back in a week or two, you probably find some and actually go outside and see it pass overhead, which is pretty fun. So we spend 90% of our time in the indoor environment. I know a lot of you have your videos off, but probably most of you are inside right now. And when I think about the indoor environment, I like to think about some of the trade-offs that we think about there. We have indoor environmental quality, which is actually the quality of our indoor environment, what we might be exposed to while we're there. And then we have a balance between energy and health. So buildings actually use about 40% of our energy through things like electrical energy as well as ventilation. And there are some trade-offs as well with health. So asthma alone costs about $81.9 billion a year, and your indoor environment can impact asthma and health. So this is kind of the system that I work in. And I'm going to talk about 3 applications of this system today. The first application is thinking about mold and really trying to characterize microbial growth in the built environment. This is really important because today exposure to mold in housing costs an estimated $22.4 billion a year. And it's usually not as pretty as this piece of artwork over here. It accounts for about 16% of the costs associated with allergic rhinitis, acute bronchitis, as well as asthma. And this problem also disproportionately affects underserved communities. And so it's really important that we understand the conditions under which mold growth occurs in the built environment and how we might be able to characterize that and then also prevent it in the future. As a specialized application of this, we also think about microbial growth in specialized environments, such as on the International Space Station. Microbial growth has actually historically been a problem on spacecraft. If you look at Mir, at the time of its decommissioning, it actually suffered from pretty extensive fungal growth. The fungi were actually degrading components like the wiring and window seals, which are pretty important when you're in a space environment. We learned a lot of lessons from designing spacecraft, and now we've done a much better job on the International Space Station in controlling the indoor environmental quality. But occasionally you still run into problems when you have this sealed environment with people inside breathing, sweating, exhaling moisture. And so this is just an example of a fabric panel where mold growth occurred in the hygiene unit where astronauts were hanging their sweaty towels. And so we can apply some of the things we're doing to better understand microbial growth on spacecraft to help identify and prevent and stop it. Finally, I'm going to talk about what's on everybody's mind and how we can think about the indoor environment and how that plays out into COVID-19 surveillance. So this is some really new hot off the presses work that we've been working on, and I'm really excited to share that with you today. So really, my talk is divided into these three parts. Like I said, we're going to look particularly at moisture and carpet. Everybody's favorite talk. I'm sure that's exactly what you were thinking about when you woke up this morning. And then we're also going to talk about spacecraft and also infectious disease monitoring, particularly with COVID-19. So the first topic we're going to talk about is modeling microbial growth in the built environment. And we're going to focus specifically on carpet. In the past, a lot of people had focused on microbial growth on drywall, but we wanted to look at growth actually within the carpet. And there's a couple of reasons for this. The first reason we wanted to look at carpet is because it's so prevalent in the built environment. You can look at a graph and see how prevalent carpeted floors are. This is a graph of the percentage of rooms in housing that had carpeting, and it's about 50%. Carpet today still makes up about 50% of the US flooring market. And not only is it really prevalent, but it's also a really important source of human exposure. We actually held a workshop at Ohio State in summer of 2019, fortunately before COVID-19 shut everything down, but we invited 40 experts from a variety of different fields, including carpet manufacturers, cleaners, as well as people from government agencies and academics from a variety of fields to come and actually talk about what it means to have a carpet in your indoor environment. So from that workshop, we put together this review that talks about the implications for carpet on chemicals and microbes in the built environment. And one thing that really came out of this is how much carpet serves as a reservoir for dust. You do get sort of more resuspension out of carpet in a room compared to a solid surface flooring. And it also retains that dust as well. So it's a really important source of human exposure. And it's important that we understand what's going on within a carpet. Finally, the third reason that we wanted to focus on carpet when we're thinking about microbial growth in the built environment is because the relative humidity in a room can actually be elevated in the carpet compared to within the room air. And when humidity goes up, you can get a lot of things that happen. You can get excess dust mites, you can change your indoor chemistry, but you can also potentially get fungal growth. And so we wanted to ask a fundamental question of, could this growth actually be influencing human exposure under typical building conditions? So our fundamental question for the first part of my talk is relative humidity in the air sufficient to support microbial growth in carpet? So for this study, we collected carpet and dust from 19 homes around the state of Ohio. I like to say I'm going to start a renovation company. We're going to come take carpet squares out of the middle of rooms. It's the new fashionable thing to do. So we go into homes like this. We cut the carpet out from a homeowner who's getting rid of their carpet for aesthetic reasons, no water damage or anything like that. We take it back to our lab, and with the matched dust samples, we expose it to different relative humidity conditions, and we look at things like microbial growth in the carpet, the rate, the function, and I'm not going to talk about it today, but we've even looked at chemical degradation occurring within that carpet. So we bring it back to the lab, and we cut out a carpet square, we'll apply the dust and embed the dust with a modified ASTM method, and put it in our chamber, which is controlled at different relative humidity levels, and basically see what happens if we actually find any microbial growth occurring in the carpet. A lot of the techniques I'm going to be talking about today are DNA or RNA-based measurement techniques, where we're actually looking at a comprehensive picture of the microbial communities as well as their function. So when we take the carpet and dust back to our lab, the first thing we noted when we first started this is that the water activity of the dust and carpet equilibrates very quickly with the relative humidity in the chamber. Water activity is a measure of water availability. It's better than total water content because it actually tells you what water is more chemically available to the microbes. It's used a lot in the food industry to say something, like, are your granola bars going to have microbial growth on them? And it's equivalent to the equilibrium relative humidity in the chamber. So for instance, a relative humidity of 85% is equivalent to water activity of 0.85. And what we notice is that in the dust after 1 day and 1 week, we get very rapid equilibration with that relative humidity in the chamber. So you're really increasing the water activity of the dust. And with this first experiment, you can see in the cup in the bottom right, we knew we were onto something because we actually observed visible fungal growth on the dust itself after 1 week at 100% relative humidity. So when we measured the microbial growth in this carpet, we observed growth occurring in the dust and in the carpet above about 80% relative humidity. And so on the top left, you can see a graph of the fungal growth. You can see spore equivalents per milligram dust on the y-axis, as well as the original dust and the equilibrium relative humidity on the x-axis. And you can see increase in fungal concentrations as the relative humidity increases within the jar. The story is really about the fungi here. Ironically, we started this as a bacterial project, but there's really not a lot of bacterial growth occurring in this sample, except at 100% relative humidity. On the bottom graphs, we continued this growth over time at 100%, 85% relative humidity. At 100%, we stopped the experiment after 3 weeks because there was just such extensive visible fungal growth on our carpet squares that we said that's good enough. Let's end it here. And we saw that growth continue out to about 6 weeks for 85% relative humidity. So with this, we were able to establish that microbial growth can actually occur within the dust and the carpet at these elevated relative humidity levels. So after this, we wanted to continue characterizing this growth and see if we could learn a little bit more about when this occurs and what this looks like. We also measured some of the nutrients in the dust, and we established that moisture is really the limiting factor for this growth. None of these nutrients that we measured were in any quantity that would be limiting. The nitrogen was maybe close to being limiting, but not really. In this sample. So it's really all about the moisture in the indoor environment. And as soon as you get enough moisture present, you can actually trigger that growth to start occurring. Finally, we used a rough model of human exposure to see how this growth might actually contribute to the total concentration of microbes that you might be exposed to. So on the left, you have the estimated concentration in the air, and on the bottom, you have your different relative humidity levels after 1 week. And in the orange, you have the background level you might be exposed to in a typical room. And then in the blue, you have the additional amount that you might be exposed to from this growth. And you can see that the growth actually increases the microbial concentration you might be exposed to. In addition, the things that tend to grow at these elevated conditions tend to be things like microbial, like fungal species that contain known allergens. And so this might be especially problematic. So in addition to this initial study, we also wanted to see if we could actually quantify this growth and predict it under varying relative humidity conditions. So this work was predominantly done by Sarah Haynes, and it was conducted in these homes across Ohio. And what we were looking at now is we recognize that not everybody has their house sitting at 85% relative humidity exactly for 1 week. Or at least I hope you don't. But what's more common that actually occurs in the indoor environment is you have someone take a shower, or there's a cooking event, or it's a hot, humid summer day, you have your windows open, and the relative humidity goes up for a short period of time, and then it goes back down. And we wanted to ask if those types of variations might be sufficient to support microbial growth in the built environment as well. So we did an experiment where we exposed these samples to variations in relative humidity conditions of dust embedded in carpet. And we had two different levels, 85% and 100% relative humidity. Both these are very high. 85% is a pretty high relative humidity for an indoor environment, but it's not unreasonable that could occur after, say, a showering event or something like that, or under suboptimal conditions. And we exposed them to these elevated conditions from 50% relative humidity for 0, 6, 12, 18, and 24 hours a day. And then we measured the resulting microbial growth to see if we could model it. We wanted to model it using the time of wetness model that was previously developed to model microbial growth on drywall. And what this does is it compares the time of wetness, the percentage of time spent above a critical moisture threshold, in this case, 80% relative humidity, to your growth rate of your microbial communities. And in blue, you have your standard model, which is basically the more time that you're at this elevated moisture condition, the more growth you get, and it's a linear relationship. And then you can have different models that predict how different changes in how the growth stops or starts might alter the quantity of growth that you get in the end. What we were able to do with this model is actually realize that we can indeed predict how much microbial growth you'll get in a carpet and dust community at these different relative humidity conditions for short periods of time. So this is the data here at 85% on the left and 100% on the right. And you can see that this fits really well with actually the activation limited growth model, or the two-stage activation limited growth model. And what this says is that when it first becomes damp in an environment, you might have a slight delay in when microbial growth actually begins. But with this information, we can actually model how much microbial growth you might get. And this allows us to do some important things, such as if you're having large models on a big scale about how mold might be affecting a large group of kids, and you want to mathematically model how reduction in that growth might improve their health, this is something that you could use there. And then you can also use it to predict how much growth you might get in certain conditions, which can be very important for different environments. Another observation from this data is that you have a lot of very site-specific effects from microbes in a given environment. And so I'm showing you this graph largely because it's kind of a mess. And what's on this graph, this is a principal coordinate graph where similar microbial communities cluster together. And the samples that were exposed to higher moisture conditions are darker, and the samples that are exposed to less moisture conditions are lighter, and each site is a different color. And what we really wanted to see on this graph, if moisture changed your microbial communities in a set way, is that you'd have a group of samples altogether that were dark and a group of samples that altogether that were light. But that's not actually what happened. The samples actually grouped much more strongly by site, by color on this graph. And so what this means is that actually each microbial community in a home is so unique that that dominates any moisture signature. This actually partially explains why it's been so challenging to develop a metric or some measure of microbes in a built environment that's consistent across samples, that consistently indicates if you have a dampness problem in a home. And a lot of the reason for that is because these microbial communities in each home are so individual and so unique that it really dominates any moisture signature that you get from the individual species. So we wanted to go a little bit deeper into this to understand what's going on, and we wanted to look at microbial function. So we actually looked at the RNA, or the gene expression analysis, and this work was done primarily by Bridget Hagerty, and we looked at different water activity levels that these microbes were exposed to and the actual gene expression. So this is a principal component plot, also where similar samples cluster together. And what we found is that the water activity changes the gene expression pretty dramatically when you get to a water activity of 1 or an equivalent relative humidity of 100%. And not only does it change the gene expression from those communities at 50% or 85% relative humidity, but there's a lot of variation there, a lot of metabolic diversity. And these samples, even though they were from the same home, embedded in the same carpet, incubated at the same time, even end up very different from each other. From this data, we were actually able to understand more about the microbial function that's happening in this built environment. And so this is my favorite GIF. I'm pretty sure it's about to go viral, so look for it on your social media feed any day soon. But what this shows is each of these lines is a different gene in a pathway that's being expressed. And you can see here that at 50% in the upper left corner, you have all these core metabolic functions that are complete. We have lipid metabolism is complete, carbohydrate metabolism is complete. At this stage, the microbes are really just barely hanging on. They don't have enough moisture to do much else. Around the outside, you start to get more of these genes showing up at 85%, and then even more at 100% relative humidity when you have more moisture. And this is really important because on the outside is where these secondary metabolic pathways are. And those tend to be in fungal species where the allergens are and the mycotoxins are and the virulence factors. And so that means that as you increase relative humidity and moisture in these environments, not only do you get potentially different species showing up, but you also get potentially more allergens. And you might have an increased response to an individual spore. So it really begs the question of, is a spore, or does it matter where that spore was actually grown and under what conditions? We wanted to see if you did actually get more allergens at these increased moisture conditions. So we did, and we looked at the gene expression of individual allergens. This is a violin plot on the left, and we did actually find substantial upregulation of known allergens in this data set. On the right, we also measured this in a protein-based assay. This had to be done in an individual species, but what we actually did find is that at increased water activity level, you did indeed get more allergenicity per spore, or more of these allergens present per spore, even on a protein basis. So this shows that increased moisture conditions also increase allergen potency, just like we saw in our functional pathway. We also looked at this again on a different allergen. Here we looked specifically at one allergen at Alt-A1, and this was work largely done by Nick Nastasi. And you can see that the Alt-A1 allergen also increases as you increase relative humidity conditions in the carpet. In this particular study that I'm highlighting here, we also looked at different factors that might influence how much microbial growth you get. And what we found is that moisture was still the most important factor, followed by how much dust you have, and followed by carpet fiber type was also important. And so we were able to once again confirm that at these elevated moisture conditions, you have more allergens that are actually present. We also took some pictures, so I'll warn you, you can't unsee the next slide. But we took some SEM images of microbial growth on carpet. And here you can see the large nylon carpet fibers with the dust and the fungal growth on top at different relative humidity conditions. And at 50%, we don't see a lot of microbial growth. We pretty much see some nylon fibers with some dust particles on top. But at 85 and 95%, we start to see hyphal extension of the fungi and that growth showing up. And then finally at 95 and 100%, we see much more extensive fungal growth and actually presence of fungal fruiting bodies showing up, which really corroborates our previous data about the increase in total fungal concentrations at these different conditions. And here we can actually visually see it under the microscope. We also took a confocal Z-stack of this, so you can actually see how the fungi actually wrap around the carpet fibers in this community. And it appears that they are mostly using the dust as a nutrient source here, but there's, in this particular situation, they're still wrapping kind of around these fibers. So in summary, for part one, we showed that elevated moisture conditions are really sufficient to support microbial growth and function in dust. So it's really all about the moisture. The moisture indicates if you get that growth, how much growth you actually get. And then finally, if you're going to have additional things like allergens present, we can actually model a lot of these things quantitatively. And this has really important implications for health and building design as well. All right, so that's a lot of the background information for actually part two of my talk, where I want to move into talking about space dust. So we wanted to ask if you could apply a lot of these things in a specialized environment, not only in houses, but also in a specialized environment like spacecraft. So we were really excited to work with NASA and receive some dust from the International Space Station, where we could expose it to different moisture conditions and then look at what is actually happening in those microbial communities and if we can model that growth as well. And on spacecraft, this has a lot of really important implications for the human health of the astronauts who are on the spacecraft, as well as planetary protection and spacecraft integrity, especially for, if we do have any long duration missions. So relative humidity is really important to control on spacecraft. We saw that with previous spacecraft, particularly with Mir. And we can see it even in the dust visually from the International Space Station. If you incubate this dust at 50% compared to 100% relative humidity, you can actually visually see a change in color after an extended period of time. And so here, as I said, on the International Space Station, we do a great job controlling it. It's very, very clean up there, but this models what might happen if you had, say, a ventilation system failure or some other unintended scenario, and what might happen in the dust up there. The dust on the space station actually looks very similar to carpet. Little did we know when we were working with carpet that we were actually just getting ready to be able to image this very fibrous dust. So we were excited that it looks pretty similar to our carpet matrix on Earth, and so that we could understand what was actually going on. So as I said, we took this dust and tried to model a ventilation system failure in the lab. So we incubated at these different relative humidity conditions, and we once again saw fungal growth occurring just like in dust on Earth. So at 85% and 100% relative humidity, you can see that this fungal growth occurred. And this is going to allow us to model what would happen under an unintended scenario, such as a ventilation system failure. And once again, we can quantify this growth at different relative humidity conditions. It looks very similar to the dust on Earth. And you can see that both the length of time that it's incubated at these conditions and the relative humidity determine the growth that's present. We're still working to take this data and do more quantitative work so we can do more predictions of the growth that would actually occur under these given scenarios. But we were really excited to see from this work that the dust on the International Space Station is actually very similar to the dust on Earth. And we can provide some important information to NASA about the different conditions where you could expect this microbial growth to potentially start occurring. All right, so I'm going to move to part 3 of my talk, and I'm really excited to talk with you all about this today. We've been doing some work in my group on COVID-19 surveillance and looking at the indoor environment as a potential source of information for surveillance of this disease. So at this point, we have the vaccine distribution, fortunately, and we're really looking forward and recognizing the fact that we're going to need long-term monitoring into the future. Unfortunately, COVID-19 is unlikely to be eradicated anytime soon, even with vaccine distribution, and so we might continue to see future outbreaks. The vaccine, fortunately, will reduce the need for individual testing, but we're still going to need long-term monitoring to understand if we have variants that are showing up or if we have individuals without immunity who might be exposed under different scenarios. And so it's important that we have really solid monitoring strategies in place to catch any outbreaks early so that we can react accordingly. Wastewater monitoring has been really successful and has been demonstrated by so many great research groups as a viable option for long-term monitoring. And the benefit of wastewater monitoring is that you can really look at an entire sewer shed to see the different, the disease prevalence in that entire community. So wastewater monitoring allows you to monitor a large population for a relatively low cost. On the other end of the spectrum, you have individual monitoring, where it's more, it costs more and is more cumbersome, but you can get more high resolution data of the low numbers of individuals monitored. We were looking to add a tool potentially in the middle of this, where you might want to do monitoring at the building scale and look at high-risk buildings such as congregate care facilities, prisons, different dormitory situations, anywhere where you're especially concerned about catching an outbreak early. And you can use wastewater monitoring at the building scale, but it can be challenging depending on the building setup and where you need to take the sample. So we wanted to evaluate if there was a better way to do this at the building scale. And when COVID-19 started, everybody wanted to chip in any way they could against the fight of this pandemic. And so we sat down and thought for a minute. We said, we're the indoor environmental quality group. We want to think about where this virus is actually going in the indoor environment and how we might be able to monitor it at the building scale. We wanted to contribute any way we could, especially with everybody else doing their part, all the healthcare heroes out there on the front lines, and see, is there a little piece of this that we could potentially contribute to? So most of the viral spread actually occurs in the indoor environment, and you can think about where the virus goes. Here's a lovely picture of a sneeze here, and you can see the different particles that are being expelled. And one thing you'll note is that there's a lot of different particles in different size ranges. You have very small particles and very large particles. And the very small particles are being suspended into the air. But if you'll notice at the bottom, you have a lot of these very large particles that are going to fall out directly to the ground. And so one thing we realized we needed to think about was where the virus is actually going in the indoor environment. And so for this, you need to understand aerosol particle size distribution. So the virus is actually present in a wide range of particle sizes. The virus itself is about 0.12 microns in diameter. It's much bigger than a VOC, so this is the size of formaldehyde over here, just for scale. But the virus is not usually floating around alone. It's usually in droplets that range from about 0.1 to 100 microns in diameter. Most of those droplets tend to be around 1 micron in diameter, kind of shown as this particle size over here to scale. And so if we understand this, we can understand the distribution of these droplets in the indoor environment. So on this graph, you can see particle settling and how this typically occurs indoors. So on the y-axis, you have deposition velocity. So particles at the top end of this settle out really fast, and particles at the bottom end settle out really slow. And on the x-axis, you have particle diameter. And you can see that the really tiny particles tend to settle out very quickly due to Brownian motion and electrostatic forces, and they're pretty efficiently removed from the built environment onto even like the ceilings and walls. Where the virus is and where a lot of these droplets are tend to be in this kind of dip in this graph. And these particles have very slow settling velocities. So they stay in the air for a really long period of time. And a lot of the particles that you exhale when you're breathing, coughing, talking, sneezing, whatever, tend to be around this kind of 1 micron size that stays in the air for a really long time. But there's also a lot of larger particles that are emitted, and these tend to settle out due to gravitational settling that doesn't affect the smaller particles as much, and those settle directly onto the floor. So we said, given this, let's think about where the particles going in the indoor environment. And viral transmission, I should also note, is pretty complicated. It's, as you can see, if you've been following the news, it can be really difficult to figure out exactly what the dominant transmission route is, because a lot of these things occur concurrently. So you can have large droplets, you can have small aerosols, you could also have direct or indirect contact with people. But we wanted to kind of understand where this is actually going in the indoor environment in this complicated situation. As I mentioned previously, one thing we knew we wanted to look at was the dust. And the reason for this is because the dust serves as a pretty effective microbial reservoir, as we had seen from our previous work on carpet. We also know that other viruses are present at high levels in floor dust, and there's been some anecdotal studies about SARS-CoV-2 on particulate matter and things like that. So we said, let's look at the dust, let's look at all these other surfaces too. So let's look at other surfaces, as well as air samples, and try to figure out where this virus is actually going in the indoor environment. And if we can figure this out, we think we can potentially add a new tool in the fight against COVID-19 to have a potentially more effective monitoring tool at the building scale, especially when it's really difficult to do wastewater sampling in a particular building. So the goal of this work was to use dust as a matrix to see if dust could be an effective matrix for outbreak surveillance. To do this, we collected samples from rooms of students in isolation who had a positive test for COVID-19. We collected bulk dust samples as well as surface swabs and an indoor air passive sampler in the built environment that consisted of a flat styrene sheet as well as some carpet samples in there to see what might be the most effective for monitoring at the building scale. One thing I wanted to be clear about is that for this, we did measure RNA, and that the RNA of the virus is very different than any measure of infectivity. If you look at the viral structure at the bottom, you have this outer shell with the glycoprotein spikes. There's a capsid inside that, and the nucleic acid, in this case RNA, is within that capsid. And so you can still have that RNA present if the outer shells have been degraded and it's no longer infectious. So in this study, we were measuring only RNA. We did not measure anything about infectivity as well, just so I was clear on that. But when we looked at the different sample types in the indoor environment, we first looked at concentration. So here you can see the results of different surface swabs, as well as the floor dust and the passive sampler. And what we saw is that using 3 different PCR-based measurement methods, the floor dust pretty consistently had the highest concentration of virus in our sample, especially compared to all the different surface swab types, as well as the passive air sampler. We also looked at the percent of positive detects. And once again, we saw that the floor dust was the most consistently positive of the different sample types that we collected. Using these different sample types, about 89% of the floor dust samples were positive from these rooms, compared to only about 55% of the surface swabs and around 20% of the passive sampler samples. If you use droplet digital PCR, one of the 3 methods we used, you can actually get 97% positive detects of the floor dust in the indoor environment. And so we were really excited about this, which indicated that the floor dust might be the best matrix to use if you're interested in doing monitoring at the building scale, because you do get a higher concentration of the virus in the floor dust compared to other sample types. And you also get a higher percentage of positive detects from that dust. We looked at the RNA concentration over time as well. So we let these bags sit in our lab for 4 weeks and to see if the RNA would decay. We were a little bit surprised that it did not decay in panel A here over a 4-week time period. And what we realized is that it's probably because the RNA is very well protected within the capsid. This was pretty surprising because if any of you have ever handled RNA in the lab, it's really, really challenging. If you literally breathe on it or touch it, it will be destroyed because of the RNases that are present, and they're also highly present in dust. In fact, one of the major challenges to doing this study was being able to extract RNA reliably from dust. If you get a RNA extraction kit that's commercially available, there's usually a warning in there. Please make sure you don't get dust in your sample because it will degrade your RNA. And our samples actually were the dust. So we had, fortunately, when this happened, been working for several years to perfect that method. But we were really surprised that the RNA in the virus did not actually show any decay over time. This does make it a little easier, though, for potential long-term surveillance because it means you don't have to freeze your samples. They're pretty stable. You can collect them, ship them out, and get them analyzed. We also looked at our 3 different methods, and these are cumulative normal probability plots in B, C, and D. And one thing we did note is that we did get a lot of variability even within samples from the same vacuum bags. The reason for this is most likely because we couldn't homogenize the dust and sieve it like we usually do due to biosafety concerns related to the virus. So we did see a fair amount of variability in our samples, and this should probably be addressed in the future either by taking multiple samples from the same bag or finding a way that's compliant with biosafety concerns to sieve and homogenize the dust. One thing that was also really interesting about this study is that the isolation rooms were sprayed with a disinfectant prior to collection of the sample as part of the normal protocol. And the disinfectant that was used is a non-selective oxidizer. It's expected to destroy protein of the virus. There's mixed literature on how it affects RNA, but even with the presence of this disinfectant within our samples, the RNA still persisted and it was still protected within that capsid. So we thought that was pretty interesting. So the next obvious question that people ask me is, so what does this mean about viability of dust, of the virus in dust? And it means essentially nothing. So I just want to make sure that everybody's clear. We did not measure the viability in this study that I was talking about today. It is something we're looking into in the future, but the work that I showed today just shows the detection of RNA from the virus and not anything about viability in the dust samples themselves. So we see this as really complementary to wastewater monitoring as we move into the future. Wastewater monitoring has a lot of benefits that you can monitor really a large or small population, and you can detect an estimated 1 in either 100 to 2 million individuals who are sick within a wastewater sample. However, wastewater monitoring can be difficult in terms of sample collection, depending on where you need to collect it from a given building. You need to know where that sample is coming from within that building, who's been in there. You also need to go through some difficult pre-concentration steps with the wastewater monitoring, and not everybody sheds the virus in their feces either. It is a respiratory virus, so pretty much everybody's going to be exhaling it, but not everybody's going to be emitting it in feces. And so we actually see these as very complementary solutions, where in some cases it might be much easier to do the wastewater monitoring on a broader scale, and in some cases it might be much easier to do the dust monitoring on a building level scale. It's just going to depend on a specific scenario. In a lot of cases, these could be used at the same time in complementary ways. So we were really excited to basically provide proof of concept for this work that you could use dust monitoring within a building as a potential long-term surveillance solution for COVID-19 monitoring. And it really falls along the spectrum in terms of what size of population you want to measure and how time and cost intensive your sampling needs to be. So this is hopefully another tool in the toolkit that we can use as we move forward to continue to fight this pandemic. So some people ask, can I get my dust tested? We actually had 2 companies who saw our preprint and were super excited about this and actually rolled out dust testing commercially. I'm not affiliated with either of these companies, but if you do know of a building where they want to do long-term monitoring, you can call them up and they can test your dust for you. So if you do want to get it tested, you can contact either of them. There might be other companies out there. These are the 2 that I'm aware of, though. So in summary for my talk, in the first part, we talked about elevated moisture in carpet and dust, and we showed that elevated moisture was sufficient to support that microbial growth and function in dust as well. There's actually a lot of important functions that are occurring. We did dig deeper into that analysis, and we were even able to show that the microbes can degrade phthalates within the dust itself as well. A lot of the work that we showed here has really important implications for design of a lot of environments, both for housing, especially if you're concerned about people of asthma or allergies being present in that housing, as well as for specialized environments such as the International Space Station or future spacecraft. Finally, in some of our recent work, we were able to show that dust is an efficient matrix for COVID-19 surveillance, and hopefully we can potentially use that going forward to continue to monitor the pandemic. So obviously, there were so many people involved in these efforts, as well as the funding foundations, which I'm incredibly thankful for. Alfred P. Sloan Foundation funded a lot of this work, as well as NASA, HUD, EPA, NIHS, and NSF. And we did a lot of the analysis on the Ohio supercomputer. I, of course, want to thank all of our fantastic collaborators who contributed to these various projects, and my pretty fantastic group of students. This is a picture of us from 2019 when we could be together, but they're really the ones in the lab doing this hard work every day. So I'm tremendously thankful to work with such a fantastic group of students. With that, I'd love to have some time and take your questions.
All right, I'll clap. Thank you very much. That was awesome. I really enjoyed that. That was really cool. Lots of good stuff. We'll open up for questions, and then I'll ask questions if anyone else doesn't chime in.
Karen, I would just like to thank you. If I ever need motivation for cleaning my house, I know that you're there for me.
There you go.
But I was really wondering, so, like we're all wearing face masks now, and a lot of us are reusing our face masks, and some of us are really good about cleaning them and some of us are not. And so I'm wondering if you have any kind of thoughts about, like how allergens might be affected in that face mask environment, which is very humid and might be, being worn for, days or even much longer between washings.
That's a great question. I think I saw a news article the other day that was talking about some people with allergies have actually found relief because the face masks are filtering out some of the like pollens and seasonal allergens and things like that. I do think if you are reusing a face mask without cleaning it, you do have a potential opportunity for microbial growth, especially if you have this combination of dust and moisture, and the mask is going to get pretty damp. So you even have the potential for bacterial growth, which can occur faster than fungal growth as well. As a lot of people have said, make sure you do clean your face masks. If it's a reusable one, wear it for a day or even half a day if it's getting really damp. I know that moisture in face masks can also affect the efficiency of the filtration. So you want to be careful if it is getting damp as well. And try not to reuse them. If it's something you're wearing it for just a minute and then you take it off, it's probably not getting like soaking wet, but it is something that you want to be keeping in mind for any mask that you're wearing for a long period of time. I think that you definitely could get some potential growth there depending on the circumstances.
I'll chime in with one of my questions. I was wondering if did you do any analyses on what types of fungi that you were finding in the carpet, as well as the bacteria?
Yes. So yeah, we have sequencing data on all of this, on all of the results that we've shown. You do see some of the sort of key players that tend to show up in damp buildings. So especially at high relative humidity levels around, 95, 100%, you get a lot of aspergillus, penicillium, things like that contain a lot of allergens. Usually in the sort of like mid-high levels, so like your 85s or so, you get some Wallemia that tends to show up quite a bit. The interesting thing about Wallemia is that in some of our samples, we've seen that it almost, if you look at it from a relative perspective, like a relative percentage, the Wallemia appears to go up and then back down as relative humidity increases. But if you actually look at the total quantitative amount, the Wallemia just keeps going up. But at these higher levels, you have these other really fast growing fungal species that just increase even more rapidly. So you have a lot of different species that just fall into different categories about how fast they can grow and what moisture levels they like. But we definitely see a lot of the same players showing up again and again.
I had another logistical question. Kumar,
okay.
Go ahead.
Go ahead, yeah. So I'm curious about, I have a couple of questions. One is curious about how you actually sampled the dust from the floor for the RNA test. And then following the question of Catherine, I wonder when people wear the mask and take it out, put it on some surface. So if they have already infected, that mask may have copious amount of, virus, and they are putting in a surface. How that is going to affect in terms of indoor environment?
Sure. Yeah, two great questions. So your first question about for the virus study and getting the RNA out, how we sampled those rooms, those were actually just the vacuum bags of the people who already had to go in and clean those rooms. As you can imagine, it was very difficult to get the approvals to do that study. It took us quite a bit of time to actually get the approval to handle those samples. And so we just collected the samples that were already being collected by the people who had to go in and clean it as is. And so we just took their vacuum bags. We did not ask them to change anything. We didn't want them to stop using the disinfectant, obviously, because the last thing we wanted to do was have someone get infected inadvertently. So we said, just do exactly whatever you're already doing. And then please give us the vacuum bags. And so that's how we got those samples for the viral analysis. If you're doing RNA work and you want to look at RNA from fungi and bacteria, we've done that in the past. That is also really challenging. And in that case, when we're vacuuming directly and it's not included, it's not in that viral capsid, you mostly just need to process it really fast and get it into a solution very rapidly that inhibits the RNases present in the dust. Your second question about the masks, I know people have definitely been concerned about virus being on the outside of masks. And so I agree, like you want to be very careful about how you're handling your mask. Try not to touch the outside of your mask, because if you are in an environment where there is virus potentially in the air, that could be there. It could get on your hands, it could get on a surface. And so, yes, I think you definitely do need to be careful about how you handle your mask at the end of the day.
Thank you. One more question, maybe a couple of more actually. So it's very clear from your talk, it says RNA concentration is not an indicator of viability of the virus. Is that I understood correctly?
Yes, yes.
So if you are taking a sample and if we have a high concentration of RNAs, how do we have any indicator to show it's a potential maybe high, relatively higher than another scenario? Or you have any other ways to kind of suggest people that they have a problem?
Yes, so a higher concentration would indicate that more RNA got there somehow. So if you have a higher concentration of RNA, there was more virus present. We just don't know the status of the infectivity of the virus, because you have some number of viral particles, and then from there you have some efficiency of which you extract the sample, you get some amount of RNA out of those. And then of those viral particles, some number of them are infectious. It could be 0, it could be a larger percentage of them. And with measuring the RNA, though, you just have no way to understand how many of them are actually viable. So getting the RNA number won't tell you anything about the infectivity of the dust. You could have a very high RNA reading and the dust could be completely inactivated in terms of any infectivity potential. But a higher concentration of RNA in the dust most likely is going to correlate with more infected people in that environment, or more people who didn't have their masks and were infected, or some higher amount of virus that got deposited there for whatever reason.
Okay, thank you. Going back to the moisture content in the indoor. So how long, I'm sure you showed some graphs that shows how long it took for the fungus to grow in the dust under, say, 90% humidity, 100% humidity. What I'm thinking is, if the humidity sustains for a longer period of time, what really happens? Is that growth is going to be really exponential, and then the whole room will be kind of a maybe. So I'm extending your study to see how worst conditions might be there in this.
Sure. Yeah. So if you just leave the room at a really high relative humidity level, you do just get exponential growth. And at some point it's gonna, run out of nutrients, but you're gonna have, if you left your carpet under those conditions, you're gonna have visible fungal growth on top of your carpet. It's gonna be a pretty terrible situation. In most of those cases, you do have to throw away the carpet. There's not really a good way to remove that extensive of fungal growth. I've heard some people talk about it for like antique rugs that are like priceless, really expensive rugs that got contaminated. But for your typical everyday carpet, you're actually going to have to just throw it out. I've heard people talk about how difficult it is to sanitize carpet. If we want to use sterile carpet samples, we actually have to double autoclave it. So we have to autoclave it one time, take it out, autoclave it again, and we'll still actually have some viable fungal fragments in there that you can culture out afterwards. It's really low, you've knocked down the communities almost entirely, but there are still some spores that like somehow find the perfect nook or cranny somewhere, and will actually persist even under those conditions. So if you get a really contaminated carpet, you do just have to get rid of it. But yes, as you extend the the time and the relative humidity, you do get that exponential growth.
Okay, my last question. So I'm pretty sure the UV lights have some influence on reducing the viral activities. So do you see any potential for rooms exposed to can be treated with UV lights and be able to get out of this situation?
I know that there are a lot of people working on viral inactivation with UV lights. I share my laboratory space with Dr. Natalie Hall at OSU, and she's doing a lot of work on UV inactivation of the viral components. So I would actually need to defer your question to her. She could give you a very thorough response about different wavelengths that are very effective. And things like that. But I do think that potentially could work in different scenarios. And I think it's still an active area of research as well.
Thank you very much. Thanks for coming here and giving us an excellent talk. Thank you.
Yeah, thank you very much for having me.
So I have another question. So if you, like we heard all about, fomite transfer at the very beginning, and then now it's like, oh, well, it's maybe just breathing the air. But from your studies, I mean, I'm really thinking like, what about just resuspended floor dust? Is that a, viable like pathway for contamination?
It's really hard to say. It's something that we're actively looking into right now. It's my guess is that the predominant transmission route is still going to be droplet emissions, that you're still going to have most people getting infected because they had droplet, aerosol emissions from one person to another, like in the air. However, it's something that I think we need to look into more. As I said, it's something we're actively looking into now. I hopefully will have an answer for you not too long from now. But I think it's something we want to be careful about, and we just want to think about what we know about viral inactivation on other surfaces. I think especially this could impact people who are in the cleaning industry, who need to go into rooms after people have been in there infected. And one thing that even the CDC recommends on their website, if you're going into clean schools, is, hey, wait some period of time before you go in there and clean. And that's not only going to allow time for the virus to inactivate on these different surfaces, but it's going to let any of these, teeny tiny aerosol particles settle out of the air. And so I think we do need to be careful about that, because the jury's still out on if that is a potential source or not.
Cool. I had just another logistics question about, so I think you said you had carpets from 19 people and they were volunteers. So did you just kind of put out an advertisement to say, oh, if you're changing your carpet, participate in the study? Or did you like actively recruit people? Or how did that work for that? Or, and they were going to be replacing it, right? You didn't say, oh, we'll replace your carpet and take out the square, or I don't know.
No, yeah, we found people who were already replacing their carpet for, and we needed to make sure it was for aesthetic purposes. We didn't want like a water damaged carpet or anything like that. We wanted sort of a normal carpet where someone was like, I don't like blue anymore, I want a pink carpet, or whatever. And so it took us at least a year to get all the carpets, and some of it was word of mouth. Anybody we talked to, hey, I'm doing this study, like if you know somebody getting rid of your carpet. So it was like, oh, I have a friend of a friend who's getting a new carpet, like, do you want that one? We did also post flyers in like libraries and different like Starbucks, Panera type locations, just anywhere that would let us put a flyer up. Hey, do you want to donate your carpet for research. And so it was a combination kind of those things.
Cool. Yeah, I was just interested in how that worked out.
Kind of a follow-up to that question. When you took this carpet in, did you take into account the thickness of the carpet and especially the age? And if so, how did that correlate to your findings?
Yeah, so the one study, I just showed a couple slides from it that Nick Nastasi did, is we really tried to drill down into the different factors of the carpet that mattered. We didn't look at the pile height, but we did from our workshop, look, like there's other studies that have looked at pile height and how that affects resuspension. But in terms of the microbial growth, as I said, moisture is really that key factor. If you have moisture, you're going to have microbial growth under most normal conditions, unless you have a carpet that's like so clean you have absolutely no dust, which really doesn't happen except for like maybe if you just bought a brand new carpet. The next factor was sort of the amount of dust that's present. And then the fiber type did also matter, but it was a less important factor in terms of how much growth you get. The other thing I wanted to mention too is a lot of people think I'm like anti-carpet, and I'm actually definitely not. I think that there are some benefits to carpet, sound reduction, injury prevention, things like that. I just think we need to think carefully about where we put it in the indoor environment. If you have someone in your home who's sensitive to or has allergies or asthma, you want to think about that. You want to think about how often can you clean it? Can you maintain it? And then also don't put it in any damp areas like a bathroom where it's going to be repeatedly exposed to these high moisture conditions, and just hang on to that moisture and that dust together. So I think there are places for it. We just need to be careful about thinking about where it does go and if it can be properly maintained.
I have one more question. Sorry. So it's very interesting. You said that the colonies are different in different houses you sampled, and I wonder whether the personal hygiene and the type of activities the people do, does it matter in terms of what type of fungus or microbes will grow, or no?
Yeah, so that's a great question. And as you said, each home is so unique. So we've done other studies looking at which factors tend to influence the microbial communities. And there are a lot of different factors that do. Some of the big drivers are geographic location. So different parts of the country or different parts of the world are going to have different outdoor microbial communities, which tend to make their way in. The amount of ventilation you use and the types of ventilation appear to make a difference, as well as occupancy, which includes both humans and pets. So having a cat or dog in your house is going to change the microbial communities that there, and even practices like, do you take your shoes off before you go inside your home? All of those factors make a small difference in terms of what's there, and then they really add up. So each house is so unique.
Mm-hmm. Yeah. Thank you very much. Thank you.
Other questions for Karen? All right, I think that Karen has agreed to stay on for a little bit longer and have an informal discussion with us. So if you would like to stay on, please stay on. If not, you can log off, and I will stop the recording now. But thank you very much for such a great talk. We were very pleased to have you speak to us today. Thank you.
Yeah, thank you so much for having me and for listening. It was great talking with you all. Great being here.
Thanks, Karen.
Yeah.